why we know mRNA works. So firstly, let's recount what we learned last time. mRNA is responsible for converting a DNA sequence, a gene, to a corresponding protein, and mRNA acts as an intermediate which is based on the DNA sequence. It then leaves the nucleus, travels to our ribosome, and is translated to a protein. So that particular sequence is what we need to replicate if we're going to produce lots of proteins. So how do we make mRNA? So a lot of the companies which make the mRNA vaccines, say for COVID, they have proprietary technology. That means it's all intellectual property and protected in that sense. So they're not openly available for us to look at, but we can make an informed decision based on what we know about mRNA and the production of it. So firstly, we need a sequence. So we either get the sequence from the protein, or we get the sequence from the mRNA coding for the protein. And in this case, for the COVID vaccine, we have a sequence for coronaviruses, and it was about finding what is different between the coronavirus sequences we had already and the COVID-19 variant that began the entire pandemic in early 2020. So when we have the mRNA for COVID, we need to extract the relevant part which codes for the spike protein. And the reason we want the spike protein is again so that we can train our immune system to detect COVID by detecting outside of this virus. We can either approach it from looking at this RNA, which is within the virus, or we can approach it from a top-down approach, which is looking at that spike protein and reverse engineering it. So the way we reverse engineer a protein like that is by making sure we isolate that spike protein on the outside of the coronavirus, then breaking it down into smaller parts. And by breaking it down into smaller parts, we can then identify what that spike protein is made of. And by doing that, we can then look back at our sequence and work out a three-base triplet or our codon. The way we analyze a protein when we've broken it down to pieces is using mass spectrometry. And mass spectrometry is a way of using the mass to charge based on each individual peptide, which is a unit of a protein, or the polypeptide chain, which is that group, a long chain of proteins as we break it down. And they act as individual fingerprints for each type of protein or that protein polypeptide chain. And by doing that, we can then slowly piece together how this protein is structured. So once we have this sequence, this DNA sequence, we can then start looking at placing it within a competent cell, such as E. coli. And by putting it inside a plasmid, which is a circular piece of DNA, we can then scale it up and replicate it and trial various different DNA sequences. And as we talked about before, if we have a DNA sequence, we can then easily convert it to an mRNA sequence. So the way we do this is that we introduce the gene into our E. coli, and we also introduce an antibiotic resistant gene too. That way we can select the populations that have both the antibiotic gene and our gene of interest. And that way we avoid culturing cells which haven't got the antibiotic gene and our gene of interest. And we make sure that both the antibiotic gene and the gene of interest are taken up in the same plasmid. Once we have done that, we can then scale and grow up our E. coli and check the expression of the COVID spike protein. And we can confirm that it is the spike protein. And this is done by adding a his tag to our gene, our protein. And that way, when we culture it up and grow it up and express it in our E. coli, we can use a column, and this his tag, which we've added to the end of our spike protein, can be captured in our Nickle column. So how do we know our spike protein is correct? How we know is by using antibodies that we know capture and react against COVID-19. So if we can create something which is similar and antibodies are bonding to it, we're onto a COVID spike protein. So what we can do with both is looking at the COVID spike protein on the virus and the COVID spike protein we made in the lab and compare them by looking at them using NMR. Well, what we can see there is local and global similarities. So are there similar groups or certain proteins in the same order, in the same chain, or in the same position? Because as we mentioned about proteins before, they self-assemble. So whilst the chains may be very similar, they may self-assemble into different ways when they reach this tertiary structure. We also can go back to that sequence and look at the similarities between the two sequences. But as I mentioned, even if they are slightly different sequences, they could end up having completely different tertiary protein structures. And finally, we can superimpose both on top of each other. And what I mean by superimposed is if we look at our hands, they're mirror images of each other. But if we were to superimpose, which means put one on top of the other, they're different structures. So you can see my thumb over here and over here. They're not in the same place. And this is very important when we want to replicate the structure. When we replicate it, we want to make sure it is the correct image and not a mirror image, as that may lead to incompatibility and antibodies that don't detect COVID. So how do we scale mRNA up? Well, the same way our cells do. We take the DNA sequence and we take RNA polymerase, put them together, and then we start producing a lot of mRNA strands. And once we start producing a lot of mRNA strands, we can then test that and verify that codes for COVID spike proteins in mammalian cells, rather than just in our competent cells which we've trialled and developed it in. And the way we do that is using chromatography and then batch processing. But the chromatography allows us to confirm that the replication steps are successful and we are reproducing the mRNA strands we require to produce this COVID spike protein. Once we've produced a lot of mRNA, we can introduce them into cells and again confirm that they are producing this spike protein. There may be replication difficulties within those cells, so we need to verify this with a few models and very rigorously to ensure that we are reliably producing this COVID spike protein. As the last thing we want is to introduce something which doesn't code correctly for our COVID spike protein. And this can take some time. Through the rigorous testing and clinical trials, we have various stages which approve and need to be approved. So one of them is confirming that the protein is the same, and that can be done using SDS page, where we compare the COVID spike protein against a known COVID spike protein is reproduced in the same way by comparing the different bands and then using the same techniques almost that we actually derive the mRNA sequence with. So by doing protein sequencing and looking at the NMR structure and shape, and then we can actually finally confirm that antibodies known to work against COVID spike protein works against the one we made in the lab. And once we have confirmed that, then it's again just clinical trials in proving that when we put it into cells, it does produce this protein, and also that there's no side effects. And with all of that, we should have a very good vaccine and something that elicits an immune response. And then it might just be tailoring it ever so slightly. So we can tailor the spike protein to be slightly broader, and that it would be beneficial so that we target any potential mutations that can occur, because we don't necessarily want a one-to-one liking to a COVID spike protein. That's so that we can have a bit of a broad range of action, so that we don't have to have repeated vaccines every year. And that should improve the immunity, so that when COVID does evolve, because it will evolve, that we don't need to constantly have to have new vaccines and new injections. So why do we use mRNA as opposed to proteins? And that can simply be that it's far easier to scale mRNA up than it can be to produce proteins to the same mass scale. And that's really beneficial when you're looking at a giant pandemic and the huge requirement for vaccines. So if we can produce mRNA very fast compared to proteins, that's always going to be a good thing. And that's not necessarily saying that protein vaccine is worse than mRNA, but if we can produce mRNA more efficiently and faster and more scalable compared to a protein manufacture or protein production, that's always going to be beneficial for us as a population. So hopefully this has been really useful to understand, and it goes into a bit more detail how we make mRNA and why we know it works and the potential future of mRNA vaccines. If you like this content, please subscribe and leave a comment below. and thanks for watching. See you next time.